Good day. Now, we will explore some major catabolic pathways that result in energy conservation in chemoorganotropes, fermentation, and respiration. Have you ever wondered how yeast ferment barley malt into beer? Or how your muscles keep working when you're exercising so hard that they're very low on oxygen? Then those questions will be answered further in this lesson. Fermentation. Fermentation is a form of anaerobic catabolism in which compounds both donate electrons and accept electrons. And the redox balance is achieved without the need for external electron acceptors. So when we say anaerobic, meaning non-oxygen requiring pathway for breaking down glucose. Organic compounds are substances that contain covalently bonded carbon and hydrogen, and often other elements. For example, sugars, lipids, amino acids, and amino acids. Next is respiration. This respiration is a form of aerobic and anaerobic catabolism in which an organic or inorganic electron donor is oxidized with oxygen in aerobic respiration or some other compound in anaerobic respiration, functioning as electron acceptors. A nearly universal for the catabolism of glucose is the Ebden-Meyerhof-Parnas pathway, or it is better known as glycolysis. This is a series of reactions in which glucose is oxidized to pyruvate. If glucose is ultimately respired, it is first catabolized through glycolysis before pyruvate is further oxidized to carbon dioxide in the citric acid cycle. By contrast, if the glucose is fermented, pyruvate is not oxidized completely to carbon dioxide, but instead it is used as an electron acceptor to achieve redox balance in glycolysis. In the series of reactions that are glycolysis, two are redox reactions. During these reactions, free energy is released and is conserved by the simultaneous production of energy-rich compounds. ATP is made from these energy compounds by substrate-level phosphorylation, a process whereby the energy-rich phosphate bond on the organic compound is transferred directly to ADP to form ATP. Glycolysis can be divided into three stages, each consisting of one or more enzymatic reactions. Stage 1 consists of preparatory reactions. These are not redox reactions and do not release energy, but instead form a key intermediate of the pathway. The first redox reaction of glycolysis occurs when glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglyceric acid. In this reaction, which occurs twice, once for each of the two glyceraldehyde 3-phosphates, the enzyme glyceraldehyde 3-phosphate dehydrogenase reduces NAD+ to NADH. In the stage 2, redox reactions occur. Energy is conserved and two molecules of pyruvate are formed. Simultaneously, each glyceraldehyde 3-phosphate molecule is phosphorylated by the addition of a molecule of our inorganic phosphate. This reaction in which inorganic inorganic phosphate is converted to organic form, sets the stage for energy conservation. Since 1,3-bisphosglyceric acid is an energy-rich compound, ATP is then synthesized by substrate-level phosphorylation. When each molecule of 1,3-bisphosglyceric acid is converted to 3-phosphoglyceric acid, and each molecule of phosphoenol pyruvate is converted to pyruvate. During the first two stages of glycolysis, 2 ATP molecules are consumed and 4 ATP molecules are synthesized. Thus, the net energy yield in glycolysis is 2 molecules of ATP per molecule of glucose fermented. In stage 3, redox balance is achieved and fermentation products are formed. The final stage of glycolysis achieves redox balance. Recall that during the formation of the two 1,3-bisphosglyceric acid molecules, 2 NAD+ were reduced to NADH. This NADH needs to be oxidized for the next round of glycolysis, and this occurs when pyruvate is reduced by an NADH-containing enzyme to fermentation products. During this reaction, NAD+ is regenerated. In fermentation by yeast, pyruvate is reduced to ethanol, or ethyl alcohol and carbon dioxide. By contrast, lactic acid bacteria reduce pyruvate to lactate. Pyruvate can be reduced to many other fermentation products, but the rationale for product formation is the same in each case. NADH must be reoxidized to NAD+ to keep glycolysis in redox balance. Alcohol fermentation. So there are two types of fermentation. This alcohol fermentation and lactic acid fermentation. Alcohol fermentation, in which NADH donates its electrons to a derivative of pyruvate, producing ethanol. With the first of these, glucose is oxidized over the 10 steps we learned about to produce 2 pyruvate molecules and 2 ATP molecules using 2 molecules in NAD+ in the process. Then each pyruvate will undergo decarboxylation to produce acetaldehyde. Finally, acetaldehyde is reduced by NADH to produce ethanol. This allows for the regeneration of NAD+, which can then be used in glycolysis again. The final product, ethanol, is the kind of alcohol that humans consume. Hence, the term alcohol fermentation, and this process has been utilized for centuries to produce alcoholic beverages such as beer and wine. And lactic acid fermentation. Again, glycolysis occurs precisely as we have learned, but this time the pyruvate that is produced will not decarboxylate, but instead it will be directly reduced NADH to produce lactate. This again allows for the regeneration of NAD+ for use during glycolysis and also involves no release of carbon dioxide. Lactate is the conjugate base of lactic acid, and it is the production of this product by certain fungi and bacteria allows them to be used in the industrial production of cheese and yogurt. Muscle cells also carry out lactic acid fermentation, though only when they have too little oxygen for aerobic respiration to continue. For instance, when you've been exercising very hard, it was once thought that the accumulation of lactate in muscles was responsible for soreness caused by exercise, but recent research suggests this is probably not the case. Lactic acid produced in muscle cells is transported through the bloodstream to the liver, where it's converted back to pyruvate and processed normally in the remaining reactions of cellular respiration.
Fermentative diversity. Not all compounds are inherently fermentable, but sugars such as glucose and other hexoses, as well as most disaccharides and other relatively small sugars, preeminently fermentable. Since glucose is needed for glycolysis, sugars other than glucose must be converted to glucose by isomerase enzymes. Polysaccharides such as cellulose and starch are also fermentable by bacteria that produce enzymes that attack the large molecules and produce sugars. There are different fermentations or types of fermentations that are classified by substrate fermented or the products formed. Here are some examples for solid substrate fermentation. Example: mushroom products. The substrate are straw and manure, and the microorganisms are agaricus bisporus. Other examples are soy sauce, cheese, and so on and so forth. And there are most popular fermented foods such as wine, beer, cheese, yogurt, and pickles. This table shows the summary of some major fermentations of glucose on the basis of the products formed. All of the organisms listed in the table, except for the bacterium Zymomonas, use the glycolytic pathway to catabolize glucose. Some of the fermentations listed in the table allow for additional ATP synthesis by substrate-level phosphorylation. This is possible if the fermentation product is a fatty acid, because the fatty acid is formed from its coenzyme A precursor. Many organic compounds other than sugars can be fermented and may or may not require glycolytic reactions. Some endosspore-forming species of Clostridium ferment amino acids, while others ferment purines and pyrimidines, the products of nucleic acid degradation. Some fermentative anaerobes even ferment aromatic compounds. Some of these cases, the formation of coenzyme A fatty acid derivatives in the fermentative pathway is key to energy conservation. Example: the coupled fermentation of ethanol and acetate is apparently carried out in the nature only by bacterium Clostridium cloyveri. These organisms are metabolic specialists, having evolved the capacity to ferment organic compounds, not readily catabolized or aerobically by the bacteria. Fermenters are ecologically important because of their role in degradation of dead plants, animals, and microorganisms in anoxic environments in nature. Human benefits of fermentation. During glycolysis, glucose is consumed, ATP or adenosine triphosphate is made, and fermentation products are generated. For the organism, the crucial product is ATP. Fermentation products, being merely waste products, must be discarded. However, fermentation products are not waste products to humans. Instead, they are the foundation of the baking and fermented beverages industries and are key ingredients in many other fermented foods such as the lactic and other acids in fermented dairy products like yogurt, sour cream, buttermilk, cheeses, pickle, certain sausages, and fish products. The benefits of fermented foods are the enzymes. Increased enzyme content helps you to absorb nutrients, reducing the need for vitamins and supplements. Probiotics. These good bacteria help restore balance in the gut and aid digestion and immune health. Safety. The lactic acid created during the fermentation process kills Escherichia coli, making it safer to consume than raw vegetables. Preservation. The lacto-fermentation process stores food longer than canning without depleting nutrients. And the nutrition, the fermentation process increases the nutritional value of enriching certain nutrients. And that's all for this video. Thank you for watching.